Today's lecture topic is spermatogenesis. The learning objectives are define spermatogenesis and what is the normal morphology of the sperm, what hormones are required for their specific role in spermatogenesis, and what is the composition of semen, and define capacitation of the sperms. Before we start to discuss the physiology, here are the physiologic anatomy. If you see here, the testes are the paired organ. Their lies are hanging down into the scrotum. If we dissect the testes, it is mainly divided into the two portions. The seminiferous tubule, these are the seminiferous tubule, and the interstitium. This interstitium is filled with the Leydig cell, as these Leydig cells secretes the testosterone. 90% of the testicular mass is just because of the seminiferous tubule. In these seminiferous tubule, these are about 900 coiled tubular structure in which sperms are going to be formed, and each one is about 1.5 meters long. So sperms are formed in the seminiferous tubule. They pass on to the epididymis. Epididymis, it lies over the tip of the each testes. So this is the epididymis. This is the tail of the epididymis, the body of the epididymis, and the head of the epididymis. This epididymis is also a tubular structure about 6 meters long. Then sperms are passed to the vas deferens. So this is the vas deferens. Then sperms are passes through and they are emptied into the ejaculated duct. Now this is the ejaculated duct which lies in the mass of the prostate gland. So this is the prostate gland. On both sides of the prostate gland, this is the vesicular structure, vesicle, and they are called as the seminal vesicles. The terminal part of the vas deferens, it is slightly dilated, and this terminal part which is dilated is called as the ampulla of the vas deferens. Then this is the prostate. This is the posterior urethra. So when the sperms are going to be formed, they pass through the vas deferens and they enter into the ejaculated duct. This ejaculated duct opened into the posterior urethra. Now this is the bulbourethral gland. This bulbourethral gland, it secretes the mucus. The inner lining of the urethra, inner lining of the exterior urethra is also lined by the mucus secreting cells. So both the mucus from the bulbourethral gland and the inner lining of the epithelium of the external urethra both facilitate the passage of the sperm through the urethra. It lubricates and facilitates the passage of the sperms. As far as the introduction is concerned, the spermatogenesis occurs in all seminiferous tubule, beginning at about 13 years of the age up to the end of the life. But the rate of sperm forming is markedly decreasing in old age. During the meiotic division, the male Y chromosome goes to the one spermatid and becomes the male sperm, and the female X chromosome goes to the another spermatid that become a female sperm. The sex of the offspring is determined by which type of the sperm fertilize the egg. So as far as the stages of the spermatogenesis are concerned, so spermatogenesis starts at the age of the puberty, just around 12 to 14 years. This is the seminiferous tubule lined by the modified epithelium, which are called as the spermatogonia. These spermatogonia undergo several mitotic division to give rise the sperms. This is the another view of the seminiferous tubule. This is the lumen of the seminiferous tubule. These seminiferous tubules, they are lined by the Sertoli cells. So this is the Sertoli cell. The Sertoli cells are having very large cytoplasmic extensions, and these cytoplasmic extensions, they overlap the subsequent cytoplasmic extensions of the Sertoli cells, and they are formed the blood testis barrier. But these sperms, they are formed and squeezed through the blood testis barrier, and then these sperms, after passing through the different developing stages, they goes into the lumen of the seminiferous tubule, and from the seminiferous tubule, the sperms they pass into the epididymis. So the different stages of the spermatogenesis, at the onset of the puberty, the spermatogonia, which are the germinal epithelial cells, they undergo several divisions. They go under several divisions, and it requires about 25 days. So after the 25 days, they are transformed into the primary spermatocyte. Then after the about 9 days, these spermatocytes, they undergo reduction division. A meiotic division number 1 occurs, in which all the 23 pair of the chromosomes, they are being separated. So one set of the 23 chromosome goes to the one secondary spermatocyte, the another set of the 23 chromosome goes to the second spermatocyte. Then after few days, a second meiotic division occurs. In which the 23 chromosomes, they split apart at the centrosome, forming the two sets of the 23 chromosomes. So one set passing to the one daughter spermatid, the another set of the 23 chromosomes, they passes to the another daughter spermatid. So spermatid, then these spermatid finally formed having the 23 chromosomes, one half of the genes of original spermatogonia, the other half of the genes provided by the female ovum during the fertilization. So formation of the sperm, after few weeks of the meiosis, each spermatid change into the spermatozoon, which is the singular form of the sperm, losing some cytoplasm, reorganizing the nuclear material to form the compact head and the formation of the tail. So the period of the spermatogenesis takes about 75 days. The mature sperm is having the two portions, the head and the tail. Now head, it is the condensed form of the nucleus. On outside, 2/3 of the head is the thick cap-like structure, which is called as the acrosome. So this is the 2/3 of the head, a very hard disc-like appearance is called as the acrosome, and this acrosome is formed by the Golgi apparatus, and this acrosome containing a very important enzyme. These enzymes are hyaluronidase, which digests the proteoglycan filaments, and the another enzyme is the proteolytic enzyme that digests the proteins. The tail is having the three compartments: the centrally lying exoneme, which is made up of the 11 microtubules, which is covered by the cell membrane, and the third is the mitochondria, a very large number of the mitochondria containing in the tail. So movements of the tail, which is called as the flagellum, provide the forward movement to the sperm, So energy for movement supplied by the mitochondria. The velocity of the sperm movement is about 1 to 4 millimeter per minute in female genital. For normal spermatogenesis, certain hormones are required. So number one hormone is the testosterone. Now testosterone is essential for the growth and division of the germinal cells. Luteinizing hormone is required indirectly because it will act upon the Leydig cells to secrete the testosterone. The third hormone is the follicle stimulating hormone. It will act upon the Sertoli cell, and it is the main hormone required for the conversion of the spermatid into the mature sperm. Estrogen is also required, essential for the spermiogenesis, and the estrogen, it is formed from the testosterone by the Sertoli cells. Because Sertoli cells are having the certain enzymes which aromatize the testosterone in order to form the estrogen. Growth hormone is also required for the metabolic function of the testes, which promotes the early division of the spermatogonia. In absence of the growth hormone, the person becomes infertile. And then comes the maturation and storage of the sperm. Sperm requires several days to pass through the epididymis, from where it develops the motility after 1 to 2 days. So motility of the sperm, it acquires from the epididymis. Remember, so several inhibitory proteins in epididymal fluid still prevent final motility until after ejaculation. Two testes form about 120 million sperms per day. Small quantity of the sperm is stored into the epididymis, major store in the vas deferens, in the terminal part of the vas deferens, which is called the ampulla of the vas deferens. Motility of the sperm increased in neutral and slightly alkaline medium. Its motility reduces in the acidic atmosphere. A strong acidic causes the death of the sperm. Activity increases with increasing temperature, but so does the rate of metabolism, which causes the shortening of the life. Life of the sperm in female genital tract, it is only 1 to 2 days. The specific function of the seminal vesicles, which are lying on the both sides of the prostate. This is the seminal vesicle, which is the tortuous tube lined with the secretory epithelium, and it will secrete the mucoid inconsistency secretion, which is rich in the fructose, citric acid, fibrinogen, and prostaglandin. So during the ejaculation, seminal vesicle empty their content in the ejaculatory duct. This is the seminal vesicle. They emptied their content into the ejaculatory duct after the vas deferens empties the sperm. Because it contains the prostaglandin, the function of the prostaglandin secreted by the seminal vesicle, it reacts with the cervical mucus to make it more receptive for the sperm movement. Number one. Number two, it causes the reverse peristaltic contraction in the uterus in order to move the sperm towards the fallopian tube. The function of the prostate is a very important function. It secretes a milky fluid, contain citrate ions, calcium, phosphate, profibrinolysin, having the clotting enzyme, and a very important hormone which is called as the relaxin. So relaxin hormone in the male, it is secreted by the prostate. Now the prostatic fluid, it neutralizes the acidity of the seminal vesicle, because in the seminal vesicle secretion there is a large quantity of the citric acid which is neutralized by the prostate. So vaginal secretions are acidic. The pH is about 3 to 4. Motility of the sperm do occur until the pH rises to 6 or 7. So prostate fluid make the alkaline atmosphere of the uterus. Relaxin hormone maintains the motility of the sperm. As far as the semen is concerned, ejaculated during the male sexual act, and it is composed of the fluid and the cells. The cells are the sperm. The fluid, 60% is seminal vesical fluid, and 30% is the fluid which comes from the prostate. The pH of the semen is 7.5, milky appearance because of the prostatic fluid, mucoid inconsistency because of the seminal vesical fluid. Clotting enzymes of the prostate act on the fibrinogen of the seminal vesicle to make the weak coagulum of the semen which holds the sperm in a deep vagina. After 15 to 30 minutes, coagulum is being dissolved by the fibrinolysin formed from the profibrinolysin of the prostate. Soon after ejaculation, sperm remain immobile due to this coagulum. Semen can be stored for years at minus 100 degrees Celsius. The capacitation of the sperm means a process that makes the sperm to enter the ovum. It requires about 1 to 10 hours. So uterine and the fallopian fluid wash away the inhibitory factor present in the semen. Sperm in male genital tract normally exposed to the cholesterol containing vesicle from seminal vesicle. That cholesterol is necessary for the cell membrane which covers the head in order to prevent the release of the enzyme. But when a sperm is in vaginal tract, it swims away from the cholesterol. So sperm head now becomes more permeable for the calcium, for movement of the flagellum and make acrosome to release the enzymes. Sperm bind to the receptor ZP3 in the zona pellucida containing the enzyme. These enzymes are hyaluronidase. It dissolves the hyaluronic acid to penetrate the granulosa layer of the ova. Proteolytic enzymes digest the zona pellucida which covers the sperm. Acrosin also released from the acrosome which facilitate the penetration of the sperm. So these are the head of the sperm which releases the acrosin, facilitate the penetration. Fusion of the sperm with the ovum is mediated by the another protein which is called as the fertilin, which is released from the sperm head. So fusion reduces the membrane potential of the ovum that prevents the polyspermy. Usually 99%, more than 99%, only one sperm enters the ova. The reason behind it, many sperm reach the ovum. If you see here, a number of these sperm, they reach to the ova. This is the ova. When one sperm penetrates the zona pellucida, calcium ion diffuses inside. From here, the calcium ions they diffuse into the ova, and because of the entry of the calcium ion, it releases the cortical granules. These are the granule containing vesicles, and these granules are being released because of the entry of the calcium ion. These granules, they releases the substance. Which prevents the binding of the additional sperm. it even causes the already bounded sperm to fall. So this is the reason that the one sperm becomes successful to enter the ova, because of the when a chromosomal enzyme digests the outer layer of the ova, the calcium ions enter into the ova, which causes the release of the granules, and this granular substance, it prevents the further binding of the sperm. Even the already bounded sperm is going to be fall away. So these are the some morphological abnormal forms of the sperm. If sperms are non-motile and physically abnormal, a person is to be infertile. Person is unable to procreate. So on the right side is the normal sperm. This is the sperm having the two head and one tail, which is the abnormal form. This is the sperm having one head, two tails. This is the sperm having a very small head. This is the sperm having a very coiled structure tail, also an abnormal form of the sperm. These sperms are having a very small tail. This is the curved sperm. So these all abnormal forms of the sperm results into the male infertility. And I think that's all about the spermatogenesis. Thank you very much.